Okay, so in this part then of the chapter, we're going to look into what is bacterial growth itself, and then what are the phases of microbial growth, and then how do you count for this type, or well, how do you calculate bacterial growth? So we're going to see different techniques. So bacterial growth is actually the process of making more number of cells. So the cells, they don't grow in size, but they grow in number. And remember that bacteria has different types of growth, and then populate a sample, for instance, so they can create colonies. And then they divide through binary fission, fragmentation, budding, or through release of structures that we call conidiospores. But most bacteria divide by binary fission, in which basically one cell divides into two, and then two into four, four into eight, etc. And then when it comes to budding, in budding you will have an extrusion of a little part of the cytoplasm and the plasma membrane, of course, of this bacteria that forms like a little oval shape or round shape extrusion as it's extruding, and then it will form a complete cell after. So that's budding. And then fragmentation, you have some cells that looks like filaments, and these filaments break along the part of the filament, and that can be, depending on the cell, it can be in the middle, it can be towards the distal end, or maybe a little bit toward the proximal end of the cell. And then there are some bacteria like Actinomycetes that they have little round structures attached to one filament, and these will be conidiospores. So these conidiospores will be released into the environment, and then they will they will find an area where they can grow best, and then they start growing. And now for the four phases of growth. So we have lag, log, stationary, and dead. We have different activities, metabolic activities of the cells happening during these different phases, and that explains how then when you are plotting a curve to measure this growth, how then you have differences along the lines in this curve. So here is an example of binary fission. Here we have a rod. This is Bacillus licheniformis. So this rod, it duplicates the DNA. So here we have one copy of the DNA and another copy. And of course, as this cell is duplicating its DNA, it needs space, so it starts elongating. And then you will start forming an indentation of the cell wall in between the copies of the DNA so that they can go into separate cells. And that will happen gradually until you close actually the two cells, or well, you close the gap between the cell wall, and then the two cells separate, as you can see from this cartoon. So little by little, you start forming this cell wall in between the cells till you complete it, and then you separate these cells. And then here is the graphical representation of this growth based on the number of generations. So the original cell will be part of the G0, which will be here. And since it's binary fission, you have to use 2 in this annotation, and 0 means that it hasn't divided. So 2 means that eventually it's going to give you 2 cells, and this one you multiply it times the number. So 2 times 0 will be how much? 0, right? So your original cell haven't give you any logarithmic of 10. So you have only that original cell. And then you can see it here. So this is the potential bacteria here, for instance, if you want to visualize it that way. And then in the first generation, in this case, the cell will multiply to give you 2. So 2 times 1, that's 2. And then here you can see it. And then these 2 cells undergo the next generation. So then 2 times 2, that will be 4. So you have 4 cells. And then these 4 cells enter into the new generation. So that will be represented by this notation. So in this case will be 2 times 2, that will be 4 times 2 will be 8. And then you go to fourth generation. So it will be 2 times 2, that's 4. And then 4 times 2, that's 8. 8 times 2, that will be 16. So you will end up with 16 cells, and you keep on going. So every time, every generation, you duplicate the number of cells. So then at the 5th generation, you have 32 cells. And then 32 cells will become 64, and then 128, etc. Now, if you leave the bacteria to grow for 10 generations, it's going to give you 1,024 bacteria. 15 generations, 32,768, and so on, until in the 20th generation you have over 1 million. So it will be 1,048,576, or a log 10 of 6.02. And last time I was telling you, what if that's perhaps food that someone's going to ingest, or maybe it's a medical instrument. So if you don't sterilize properly the instruments, and even if you leave one bacteria, and that bacteria multiplies every 20 minutes, maybe by the next time, the next day, when these instruments are going to be used, they will have high numbers of bacteria. So then it is then understandable that the process of sterilization has to be very stringent. You cannot leave to chance that you have a single bacteria in your instruments or in the food. So what is generation time? It's the number of minutes that takes for a bacterial cells to duplicate. Most bacteria has a generation time of 20 minutes. Some, they take longer to divide, like Mycobacterium tuberculosis, but for instance, E. coli, the average time of replication or generation time is 20 minutes. So basically, every hour, we have 3 generations. Because it's 60 divided by 20, that's 3. In 10 hours, you have 30 generations. So then, 20 generations will be almost 7 hours. So in 7 hours, one single bacteria contaminating a medical instrument can become 1 million. And then it takes the 21st generation for that number to become 2 million. So see then how it's important to take care of a sterilization process. And then the number of generations is the number of times that the original bacteria has duplicated. So one way to calculate generation time is divide multiply 60 minutes times the number of hours that you let this bacteria to replicate, and divide by the number of generations. And that gives you minutes per generation. And then one way to calculate the number of generations is to subtract from the logarithmic number of cells at the end from the one at the beginning by this index number, which is 0.301. So then if you want to calculate the number of generations, for instance, from the previous table, like this will be your ending point, and then you can subtract this ending point from this, or maybe from this, depending on how many cells you had at the beginning, and then divide that by 0.301. And here is the representation of the different phases of growth and what happens in each of them. So this graph in the y-axis tells you the logarithmic number of bacteria, and then in here it tells you the number of hours that they have gone. So it goes from 0 to 10. And then in here, the logarithmic scale will go from 0 up to, depending on how many logarithmic scale you had at 5 times, or number, at 5 hours. So what happens in each of these phases? So if you put bacteria to grow at time 0, and then you let it grow, let's say, for an hour, and then you start measuring with a device that measures the bacterial growth, like a spectrophotometer, you don't see any growth of bacteria. The reason for this is because the bacteria starts acclimating to the new environment. It finds an environment rich of nutrients, and it starts absorbing part of the nutrients, but still is not ready to grow. So that's why the lag phase is represented by a line that it doesn't change your initial logarithmic number of bacteria. And then once the bacteria acclimates, then it will start growing in an exponential manner, and then it will reach a peak. So this is the log phase, or logarithmic or exponential increase in population. So this is where you have, in the case of bacteria, reproduction by binary fission, and if it was yeast, by mitosis, because it's an eukaryotic cell. So then, let's say this is 4 hours after you have added the bacteria into a new medium. So then at 4 hours you reach the peak, and then between 4 hours and 6 hours, you had no more increase in the numbers. So this is the stationary phase. So this is a phase in which you have the same number of deaths and the same number of growth of bacteria. So because you don't have more nutrients, you don't continue the logarithmic phase, but you still have nutrients enough to keep some bacteria growing, but some of the older bacteria start dying. So then you reach this plateau, no more growth. And then at the beginning of the next period, which is the death phase, you have decreasing number of bacteria in an exponential manner, or logarithmic rate, just as the log phase, but it's inverse. So instead of increasing the population, like in this case, you have decreasing of the population. So let's say between 6 to 10 hours. The reason for this is, well, because you lost the nutrients, and as the bacteria is dying, and you have accumulated also in here, part of the metabolism of bacteria, some metabolites, like some byproducts that can be toxic, that increases or accelerates the death rate because you have a lot of waste and you don't have more nutrients. So unless you keep adding new media, you will undergo all of these phases: lag, log, stationary, and dead. And then here is an example of how single bacteria can form a cluster of bacteria. In this case will be a Staphylococcus. Any questions? Now let's look at then how we count the growth of bacteria. So we have two major ways to do it: by direct methods and by indirect methods. So we have indirect methods: plate counts, filtration, most probable number, in which I leave you some information in there, and direct microscopic count. Indirect methods will be measurement of turbidity, metabolic activity, and dry weight. So for plate counts, we have the spread plate technique and the pour plate technique. We will see details about this. So these techniques are very useful when it comes to counting number of bacteria, for instance, in food, when you're testing the presence of microbes in food. You can grind, for instance, a sample of food together with saline solution or sterile water, and of course that saline solution has to be sterile. And when you do that, you are homogenizing the sample, and then you will grab part of the sample and either put it inside the base of a Petri plate without agar, and then you put agar that is melted, that is not too hot to kill the bacteria, or you can put it on top of the agar and then you will spread it. Now, so plate counts are usually used when you have very little sample. Now, in filtration methods and most probable number, you use these techniques when you have a large sample. Like in filtration, for instance, if you have large volume of water that you want to test for the presence of microbes, which is often done by the city labs that we have for testing the quality of water, well, you have to filtrate a large volume of water and then see if they have bacteria. So then, once you have your filter where you have passed the water, you can put your filter on top of an agar and then see if you have growth. Now, in most probable number, basically, it's called most probable number because it's a statistical estimate of what potentially this sample can have. And you use it as well when you suspect that the concentration of bacteria in your sample is low for direct plate counting, like the previous ones that we did, or when your samples have high levels of solids, like wastewater, it's an example, or soil. The limitations is that it's not an exact count, but it is good enough, like 95% of confidence interval has, that it is important to do it in certain cases. And it requires longer incubation time, like 24 to 48 hours, so you don't miss any bacteria that might be growing in your sample. And then when it comes to this indirect method, so we have turbidity. In turbidity, you have your broth, and then you put part of your broth in a little mini test tube that you insert in an spectrophotometer, and then you have a control that you also have added before into your spectrophotometer. You blank this reading from your spectrophotometer after you have added your control, and then you see how much light passes through your sample. The greater the turbidity that it is, the greater the number of bacteria you have and the less light shines through. And then for metabolic activity, well, sometimes it's easier to count byproducts produced by bacteria to indicate the number of bacteria you might have. So the more number of these byproducts that you have, taking into account that there is not a problem with the metabolism of bacteria, the more number of bacteria you suspect. And for dry weight, it's basically incinerating a sample of bacteria and calculating how much dry weight you had after. So for plate count, you use Petri plates to calculate the original number of bacteria from your samples. So you have, again, two types of techniques, pour plate and spread plate. In pour plate, you will have an empty Petri plate. You will add your sample in the middle. Then you add on the top the melted agar, swirl it, let it solidify, and then you let it grow. This technique, it's useful for samples that are not heat sensitive. And it gives you, in a way, a more uniform growth within the agar than the other technique. And in the case when you have a temperature sensitive sample, like if you're testing, for instance, a sample of food that is rich in amino acids and then you don't want to destroy them, in this case you use the spread plate method. So this, again, you add on top of the agar that is already solidified your sample, and typically the sample is 0.1 milliliters. And the reason for this is because this agar is already having a lot of content of water. So if you add more than that, the water won't absorb as easily, and it will take a long time for you to set it in the incubator. So use a small amount. So then many times when you suspect that your sample it has high number of bacteria, you have to dilute your sample to be able to count the number of bacteria accurately. And it can be coming from a broth, or it can be coming from a sample of liquid food or solid food that you grind with this saline solution, sterile or sterile water. So this is how it works with this technique. So from an original inoculum, let's say that you have a broth. You transfer from that broth 1 milliliter of broth that has bacteria, of course. It's a culture broth. So then you add 1 milliliter into this test tube that has 9 milliliters of broth. Okay? And then after you have transferred 1 milliliter of your original inoculum into 9 milliliters of broth, you have total of 10 milliliters in this tube. And then since you have added 1 milliliter into 9, and that total comes up to 10, you have something that is called 1:10 dilution. And then in this case, they show you that from this dilution, you have added into the top of the agar, which is the top view of an agar plate, you have added 1 milliliter. And after you grow this in the incubator for 24 hours, you end up with many colonies that are hard to count. Now, when you plate that amount into this Petri plate, you label your Petri plate with the inverse of the dilution, which will be 10 to the minus 1. And now, you will be doing this transfer of sample really quick before you plate, of course. And then you'll be transferring each time 1 milliliter, but you can transfer less amount or more, and then in that case, it would change the ratio of the dilution, but in this case, it's always 1:10. So then from your 1:10, you mix well your sample, and then you grab 1 milliliter, transfer it into this test tube, and then you swirl it well, and then you transfer 1 milliliter to the next tube, and always you have 9 milliliters of the dilutant. So that will be a 1:10. So then this one dilution, and this 1:10 dilution becomes 1:100, and then 1:1000, 1:10,000, and then 1:100,000, 1:1,000,000 if you keep going and doing it. But every time you dilute your sample 10 times. So what do you expect then, after you have plated on each of these plates 1 milliliter, is that theoretically the number of bacteria will be divided diluted 10 times every time. So then for your plating, this will be the 10 to the minus 1, this will be the 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5. One way that you can think about this is by basically counting the number of zeros in here. So here you have 5 zeros, 4, 3, 2, and 1. But this is the inverse of the actual dilution. So then, this one has a lot of bacteria, it's hard to count, this one as well. And then in here, in this 10 to the minus 3, you have actually 54 colonies. These 54 colonies you can count. And then if you apparently are dividing every time 10 times the number of original colonies that you have, if this one had 54, how many do you suppose to have in this previous one? If you divide it here from this 10 to the minus 2 to 10 to the minus 3 by 10, then you have to do the inverse, multiply, right? So then 54 times 10 will be what? 5,400. You just add the 0. And then for this one, multiply times 10, 54,000 colonies. That's theoretically. As you transfer and you're doing dilutions and you transfer sample, of course there can be mistakes in grabbing enough volume, etc., or adding into there. But that's what in theory it's supposed to be, but all you know that this one you can count and this one you can't. So this will be, anything that you cannot count is called TNTC, too numerous to count, or TMTC, too many to count. And then the next plate where you might have, like, let's say in this case will be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. You have 16 colonies. And then in here, you have only 3 colonies. So as you can see, I mean, you will expect that in here you have 5 colonies at least, and in here you will have 0. But then you will see then that you may have some variations, and typically when you do this technique, you make a duplicate of your plates so that you can do an average. And you have to count anything that is between 25 to 250 colonies. Anything below 25 colonies is called too few to count, or TFTC. So that's your limit. You can count only between 25 to 250. And then when it comes to counting, hopefully you don't have the extreme, something that is close to 250 or something that is close to 25. Hopefully you have something in the middle that makes it more accurate. Now, what if you have, like, for instance, 28 colonies in one plate, and in the other one, you will have, like, let's say 180. So what plate do you think is much better to report? The one with 25 or the one with 180? So let's say that in your 1 to 1,000 plate, or your 10 to the minus 3, you have 180 colonies. And then in your 10 to the minus 4, you have 28 colonies. And then you know that you need to count plates between, that has colonies between 25 to 250 colonies. So can count. For what? 180, you will pay. Why? And that's you. Someone else might have a different answer, but why?
Because it falls within the range of the...
Falls within the range, which is this.
Yes.
This one falls within the range too, right?
Yes, but the formulation is different.
The what?
The formula, the minus 3, minus 4.
Okay, let me change it for you. This is a 10 to the minus 5. This is a 10 to the minus 6. All you know that any plate, no matter what is the label, as long as it has colonies between 25 to 250, you count them. So which one will you pick? 28 colonies to count or 180 and report it? Because you have counted both of them, you know the numbers. At which one will you report? If someone will ask you, well, here is the sample of chicken. Here is a sample of fried chicken, frozen fried chicken. You study it, and then you saw that in one plate you have 20 colonies, and the other plate you have 180. And they ask you to report the numbers. Will you pick this number, the higher number, or the lower number? And why? You work for FDA.
Because the higher number has fewer, less areas, like the margin error for counting, and the lower number has a higher area of margin for errors of counting.
For counting itself, I kind of getting into what you're saying, but counting itself for me, 28 is much easier. I can count much easier 28 than 180.
Yeah, but don't you have to continue with the dilution? Or this is like final dilution?
That's all.
Oh.
For me, it's easier to count 28 than 180. And let's say that I didn't make a mistake. But which one would you report?
The higher number?
You would report the higher. And why would you report the higher?
Because to stay within, I don't know, I want to say still to stay within the range. I'm not sure why I'm thinking that, how to explain that. It's more valid, I guess.
That's the.
So
I mean,
it has a higher validity.
Both of them are within the range, right?
Right. But the 180 is more valid than the 28.
Yes, it's closer to the middle between the range,
Yes.
right?
Yeah, because the 28 is closer to the 25. So what if there was an error in the midst, but then you have the 180, and so it's closer to the middle?
Yeah.
So then you report the one closest to the middle versus the one that would have a higher rate of error or missing count or something. It makes sense in my brain. Am I making sense?
So the median is like around 112, right? So the number that is closer to the median the manufacturer will be happy if you pick that. But the public, the one that may have problem if you report the lowest.
Well, and it's probably better for treatment purposes too, no?
Mm,
Or does it?
well, in this case, it's food, so.
Oh, it's different. Yeah, sorry. Yeah, we're counting something. Yeah. I'm thinking of like strep.
So then always when you find something that is not closer to 25 or very close to 250, then pick that, and then you will have a duplicate. So you may have, for instance, in your other plates, you may have 10 to the minus 5, let's say 200 colonies. And then in your 10 to the minus 6, let's say that you have 40. Then you add this, so this will be 68, and then divide by 2. So that will give you what? 34. And then this one will be 380 divided by 2. How much will be that? I think so.
Yeah.
So when you have duplicate, then you have to do your average. So, in the previous example, where you have your 54 colonies in your 10 to the minus 3. Well, the formula tells you to calculate the number of colonies, to multiply that to the reciprocal of the dilution, and that will give you the number of bacteria per milliliter. So in this case, if you have in the 1 to 1,000, you multiply 54 times the reciprocal of the dilution times the 1,000, that gives you 54,000 bacteria per milliliter of sample. And in that case, you reported as 54,000 CFUs, or in scientific notation, will be 5.4 times 10 to the third CFUs. Some bacteria form clusters, like in the case of Staph aureus. So that's why you call it colony-forming units rather than bacteria, because out of that you can form a colony. And each bacteria, of course, comes from a single colony. Sorry, single cell. So here is then the two different types of methods. So you have here pour plate on the left and spread plate on the right. So more than likely, in the previous examples that we saw, like here, they were using this pour plate technique because they're transferring 1 milliliter of sample. In that case, it's fine, because in this technique, if you see here, you can put more sample. The sample goes in the bottom of the plate. Like this is an empty Petri plate. And then you have melted nutrient agar. You put it on the top of the sample, you swirl it, and then all the bacteria distribute within this media, and then some bacteria will grow on the top, and some kind of in the middle. So this is a nice technique. Again, it gives you more control over the actual distribution of the colonies, so that colonies don't grow on top of each other. But again, what if your sample is heat sensitive? So in that case, you don't use it. And you can use as well very little fluid, like 0.1 milliliters instead of 1. Now for the spread plate, again, you already have this solid medium. So in this case, you have to add very little fluid, 0.1 milliliters of your sample. And then with these spreaders, you can distribute your sample by rotating it, either the plate or your spreader. And then your colonies will grow only on the surface, but not in the middle of the agar plate, or the agar, sorry. And then you start counting. Similar period of incubation, 24 to 48 hours. Any questions? So we'll be doing a spread plate method. For filtration, you have a big sample of water, you have some tubing or any other liquid. It can be, actually it can be wastewater, for instance. And then you pass it through this filter. So you can have a carboy like of 20 liters, for instance. It has some tubing that is connected to a pump, And that pump is going to bring the fluid into a filter. So you have tubes connected to the filter. So you have an influent, effluent. And then you remove the filter from the cartridge where you have the filter. And if you have bacteria that is, of course, bigger than the pores, so this is like a scanning electron micrograph view of the top of a filter. And these little dots that you see here are the pores of the filter. So if the bacteria is larger than the filter pore, then the bacteria will stay here. Like in this case, this is E. coli. And then you put this, like you take away that filter, and then you put it on top of a Petri plate, and then you let it grow for 24 hours. And if you see the growth of colony, you start counting them, of course, and that tells you then what the number of bacteria that you have and the type of bacteria that you have. So there is a specific medium that can tell you when bacteria is of a specific kind. So there is these reports where they tell you the number of coliforms. And what does that mean? Well, it means that it's a bacteria that actually lives in the intestines, not only of humans but also animals. And typically when water samples that are used for human consumption have coliforms, typically the contamination comes from humans. So then that's how it is done. Now for this most probable number, what you do is dilutions, and then this series of dilutions are inoculated with these samples. So in this case, you have 10 milliliters, and then these samples are let to be incubated for a period of 24 hours, for instance. And your broth that it was initially sterile, now it has bacterial growth. And then you can see a change in the color of the medium. So in this case, all became positive. They were like this, yellow. So all became positive, so that's your first set. You have 5 positives. In your next set, you transfer from this to here 1 milliliter. Only 3 out of the 5 grows, so that will be 3 positives, so you report the positives, not the negatives. And then look in this, so you transfer from these samples 0.1 milliliter to the next, so you are diluting your samples. So how many in the bottom row grow positive? One. So then that's your total number, 531. So we have a table here that tells you the combination of positives in the first dilution, second dilution, and third dilution. So you go and find where is your 531 as this one, 5 positives, 3 positives in the next dilution, and 1 in the last. And then you go and find what is your potential number. So your lower count can be 34 and your higher count can be 250 with an index of 110. And again, you use this when you believe that you have very low counts of numbers of bacteria, or when you have a lot of solids. I mean, think about it this way, in this technique, or in this. If you add like a sample of wastewater, where you have a lot of debris, don't you think that all of these debris are going to interfere with your technique? First, you won't be able to homogenize if you use the spread plate, your pour plate. You're not going to be able to homogenize well your sample because you have a lot of solids. and then you end up with a bunch of bumps in the other. So that makes it inaccurate. And in this one, imagine if you will be able to spread really well your sample with the spreader in the spread plate method. So you want. So in this case, then you use this, the most probable number. So those were most probable numbers, spread plate, and four plate technique. Any questions about those? Now let's move then into a fast direct method for counting bacteria, which is direct microscopic count. In direct microscopic count, you have a special glass slide that has a cover, which is a cover slip. And this cover slip on the top has grids. So it has like these little squares and then these large squares. That is the area that will be used to calculate the number of bacteria you see when you put your sample under the microscope. In order to do that, your slide, which it has these little channels, will have to be first covered with this cover glass or cover slip, and then with a pipetter on the sides, like this is the top view of this device or this instrument, and this is the side view. So here you have the cover slip, and this is the glass slide with these little tunnels. So with this pipetter, you grab your sample, and then you put it here, in between, on the side. And then your liquid is going to go, it has to be a liquid, it has to be between the cover glass and your glass slide. And then you will look under the microscope, and then you start counting the number of bacteria in a large square or several large squares, depending on what are your instructions. But each large square in here represents a very small amount of milliliters. So it's 1/1,000,000 and 1/4 of a milliliter. So it's very, very tiny amount. But then you will count how many cells you have, and these cells have to be perfectly between these dark lines that makes this large square or this grid. Anything that is in between, you don't count. So in this case, you have 14 bacterial cells. Now, 14 bacterial cells, you multiply times this amount, and that gives you 17,500,000 cells in a milliliter, which will be, let me write it down in there. So that would be 1.75 times 10 to the 6 cells per milliliter. So, when would you use this technique? Well, this one is highly used in the food industry, in dairy specifically. Whenever they pasteurize milk, and then they are going to send it for human consumption to the stores, before actually they put in the gallons or the half gallon or pints containers, they have to make sure that you have a specific amount of bacteria that's supposed to be there after pasteurization. Pasteurization is not the same as sterilization. In pasteurization process, what you do is kill microbes that can be pathogens, but you don't kill all the microbes. So milk has a particular number of bacteria that is called thermoduric, and these bacteria can spoil the milk. It won't cause a disease, but it can spoil the milk right away. So I don't know exactly what numbers they handle, but let's say that this is not the number that they expect. Do you think it will make sense for the owners of these dairy farms to send, I don't know, thousands of gallons to sell in the superstores or in the stores for the public, if within 2 to 3 days the milk is going to get spoiled and they're going to have a lot of returns of milk into stores before the expiration date? It doesn't make sense. You will lose a lot of money. And not only is money, it's your reputation as a company name. So then they have their own quality control, they know how it is. And this technique, it doesn't take less than 5 minutes. It takes like 3 minutes to do this, to count and to report. So then if the numbers are high, they say, well, you know, pasteurization process failed, let's get rid of this milk. Okay? So that's direct microscopic count. And then this is what we just saw. So basically it's multiplying the number of bacteria times the volume of this little square, and that gives you the number of bacteria. Now for indirect methods, so you have dry weight, in which you basically have your bacteria that has been dehydrated or sometimes incinerated, and then you calculate the number of grams of ash that you may have or the powder that you have after dehydration. And then you would have metabolic activity method. Some bacteria may produce certain acids. You calculate the number of acid molecules that you have, and then you can have a discount and compare to a standard, in which you know how much certain bacterial numbers produce, and then calculate based on that. I always compare this as when you live in a neighborhood where people, of course, take their trash in a can into the sidewalk, and then you know that the family next to you, that house has a lot of members, and every time the trash can, the lid doesn't close because it's a lot of family members, they consume a lot of goods. But then you live next to them, and the family is very small, and then you don't consume a lot of goodies, so your trash can is never full. So similarly, you can know the number of bacteria based on how much waste the bacteria produce. The higher the number of waste, the higher the number of the bacteria. Okay? And then for turbidity method, so again, in this method you have an spectrophotometer, you have a sample that you have in a cuvette, that's like the little tiny test tube that you introduce, you have a blank, which is your control for clearing up the reading of the previous reading that you have in your spectrophotometer. Then you add your sample, and depending on how much light passes through your sample, it is how much bacteria you have. So that passing of light, it is called absorbance. And then the more turbid the bacterial sample that it is, the less light goes into this detector in this machine, and then you compare to a standard, and then you can calculate the number of bacteria. So this is how it looks internally. So this is an spectrophotometer. And then you have reading of transmission of light from 0% to 100%, and absorbance from 0% to 2%. The greater the transmission, the lesser the absorbance of light. Absorbance is how much light goes into this light-sensitive detector internally. So you have your cuvette that you introduce within this device. It has like a little slot where you put it. It has blank. So everything, it has to be 100% transmission and 0% absorbance. So when you put your bacterial sample you brought in that cuvette, all these cells that are in here will interfere with the passing of light, and only a certain amount of rays of light will go into this detector, and that can be seen in this reading. So in this case, we have 20% transmission, so 80% didn't cross. It wasn't detected, and that corresponds to this 0.20, 0.70 absorbance. That's all for this chapter. Do you have any questions?